Welcome everyone to my webinar, From Neural Recording to Functional Insight, Accelerating Discovery with Inscopix Integrated Workflow. Today, I will walk you through some of the fundamental workflow steps and the type of scientific support you can expect from my team to complete a successful project using one of our Miniscopes. Let's start with an overview of our product that we offer. At Inscopix, we've been the leader in Miniscope technology for over 14 years. We specialize in making imaging during sleep behavior as intuitive and user-friendly as possible. We are present in over 800 institutions around the world, and we have over 350 publications so far using our technology. A key part of the Inscopix experience is the ecosystem of products that we offer. These are designed to seamlessly work together to make Miniscope imaging a turnkey experience. As of today, I will showcase these products and the Inscopix complete solution from end-to-end workflow. We have the original Miniscope nVista, optimal for single-channel green sensor imaging, the nVoke for single-channel imaging with optogenetics, and the nVue for dual-color imaging. Recently, we have added to our portfolio the Quartet 2.0 for multi-region imaging and the nVista 2P for higher resolution imaging. Most of the workflow steps I will describe today are common to all these systems. In fact, these systems can be integrated into a larger portfolio of products to make imaging more streamlined. Besides the Miniscopes, we offer products that support a broader imaging workflow, such as a behavioral camera, commutator for cable management, green lenses and reagent kits for gaining optical access, easy-to-use software for data acquisition and analysis, and scientific support for the Inscopix community, which includes a team of dedicated field scientific consultants to help accelerate progress on experiments. I would like to introduce this team. Some of you might recognize familiar faces in this picture. However, my primary objective here is to emphasize the depth of our collective expertise and highlight key metrics that set Inscopix apart from other organizations. The FSC team was established almost 10 years ago and now consists of 7 members globally with top university training in neuroscience and technology. We created our learning platform, IQ, drawing on our experience supporting more than 600 laboratories, and our consultation service and workflow improvement led to a faster publication timeline. And again, we have more than 300 publications out now with our technology. By leveraging the product ecosystem, as well as the consultation service, we have successfully reduced the barriers to image. These results in the creation of more efficient workflow, introducing solutions that minimize the steps required to obtain data. For instance, the integrated lenses and the lens coating kit have reduced surgical steps from 3, as you see up here in a traditional workflow, to 1. User-friendly software for custom imaging and analysis, so allow for consistent data collection, and compared to a traditional workflow, where it might take more than 6 months to get preliminary figures, with our optimized workflow and the help of our consultants throughout each steps, you can expect to have a preliminary figure within 6 weeks from the first surgery. Now, let's review in more details a fundamental step during experimental planning, such as selecting the green lens and in the next chapter, the fluorescent indicators. At the end of these 2 sessions, you should possess sufficient knowledge to achieve a successful experimental preparation suitable for imaging with our Miniscopes. During the experimental design, your consultants will assist with the lens selection, as well as strategies for visualizing your target cell population, using either dynamic or static indicators. These essential steps are consistent across all our product line. So once you have established the optical axis for your initial experiment, you can apply this principle to develop more advanced experiments, such as multisite or multicolor imaging, or incorporating an opsin, depending on what Miniscope you have available and what is your experimental question. Our miniscopes are engineered to pair optimally with the green lenses, enabling imaging in deep brain regions. The optics of green lenses rely on radiant gradient in refractive index to guide the light rays, producing an inverted image at 1:1 magnification. As a result, these lenses are manufactured with fixed lengths and must be cut precisely either at 1 pitch or half pitch, as you see here. So to select a lens based on your brain region of interest, the first step is to identify the relevant coordinates using an atlas, referencing literature, or consulting previous experiments done in your lab. Typically, the shortest lens that achieves the required depth is preferred. However, the difference in lens diameter should also be considered, as larger diameter lenses for very deep brain areas may damage the tissue. Additionally, please account for the miniscope attachment. As you see here, this is for the miniscope and this is for the Quartet. And those takes about 1.3 millimeters from the total length of the green lens and must be subtracted by the length of the green lens. As an example, I show this from our IQ. And here you can see that a 4 millimeter lens is suitable to image a brain area located at minus 1.7 millimeter from the skull. In fact, you have 1.7 millimeters in the brain, 1.3 millimeters taken by the cuff, and then about 1 millimeter, which is optional, to glue the lens with a cement to the skull. In addition, in IQ, you can find a lens reference table where there is a summary of the most commonly used lenses in the reported and published study for each brain areas. If you still have doubts about what lenses to use, I encourage you to talk to your consultant. For first-time targeting of specific brain region or to rapidly validate coordinates, you can get practice probes. They are metal pins designed to mimic a green lens in both length and diameter. The primary advantage of these probes is that you can perform histology very quickly after the surgery, allowing for quick adjustment of targeting if necessary. They're obviously very cost-effective also compared to the green lenses.